So a lipid is an amphipathic molecule, and what that means is that it has parts that like water and parts that like oil. And so oil is technically a lipid, and lipids are present all throughout the body. Every cell is surrounded by a lipid membrane, and this consists of two very thin layers of lipids, and these help sort of form a protective shell around each cell. And proteins in that lipid membrane control what comes in and out of the cell. And so this is important in cancer because a lot of cancers show dysregulated lipids, and so you'll get certain lipids that go up and certain lipids that go down in ways that they shouldn't, and that can affect how the cells work. As I said, the lipids, we know that they change in cancer, and we know that different types of lipids can go up and down. We also know that the membrane proteins inside the membrane are really important in terms of signaling and telling cells when to grow and when not to grow. What we don't really understand is the connection between those two, how the lipids might affect the membrane proteins. And so we're trying to develop new technologies to figure this out. And so the way that we do this is that we make a little lipid nanoparticle, and inside of that lipid nanoparticle, we put the protein that we want to study. And then we have another lipid nanoparticle that doesn't have the protein. And then we slowly let those lipids exchange between the two. And so if a lipid likes the protein, if the protein binds that certain lipid, what we'll see is when we separate the two populations apart, we'll see more of that lipid in one population than the other. And so by basically quantifying using a technique called mass spectrometry, how much lipid is there in each type of lipid nanoparticle, we can say how much the given protein likes that given lipid. So if we understand that maybe in cancer a certain type of lipid goes up, and that's causing a certain type of growth factor to get activated that then causes cancer to metastasize, maybe we could develop either pharmaceutical approaches that would block that type of lipid from being synthesized, or maybe we could use things like diet to just change the lipids that people are eating and in some way affect how that receptor functions and then potentially affect how cancer progresses. The goal is basically that if we can understand which lipids are interacting with a given protein, then we can start to try and piece out what those lipids are doing. And if we can understand that certain lipids activate a receptor or suppress a receptor, that would then give us a way to start disrupting that interaction.